We will continue our discussion about fatty acid oxidation pathways that affects or that helps in the differentiation of human embryonic stem cells to the definitive endoderms. So in the previous video we have understand that it's the acetyl-CoA concentration within the cells that helps in the differentiation or that helps or upregulates this fatty acid oxidation pathways and thereby helps in the differentiation of the human embryonic stem cells to the definitive endotherms. So how this acetyl-CoA differentiate in this experimental results they have find out uh, what acetyl-CoA cause to upregulate this fatty acid oxidation. So from their initial hypothesis, they think that acetyl-CoA acetylated the proteins and that helps in the de-differentiation. So they need to find out what are the proteins that are acetylated in this uh, high acetyl CoA concentrations of the cells. So they used different conditions such as they use the embryonic stem cells without differentiated and the differentiated or embryonic stem cell that is definitive endotherms and the definitive endotherms with the chemical inhibitors, and they can see that there is a decrease in the acetylated proteins in the when the chemical inhibitors is used compared to the definitive endotherms cells or compared to the control cells, and they can see the single band that correspond to 55 kilodaltons, and this 55 kilodaltons is also the molecular weight of SMAD2 and SMAD3 proteins. So they next they made a conclusion from this Western blot, or they made a new hypothesis from the Western blot that it's the SMAD3 pathways that get acetylated, and then nucleolocalization of the SMAD3 pathways into the nucleus that helps in the differentiation of the human embryonic stem cells to the definitive endotherms. And in order to validate their hypothesis, what they did is they did the immunostaining of the cells with immunostaining of the cells to verify whether the acetylated SMAD2 and SMAD3 pathways got affected whenever the acetate is added along with the chemical inhibitors. As this is the conditions where we have seen in our previous videos that this is the condition that gives the highest differentiation. So in this case, they can see that the acetylated SMAD2 and SMAD3 in the conditions where only the chemical inhibitors are used is low. That is, chemical inhibitors reduce the acetylated SMAD2 and SMAD3 proteins. Whereas when this acetate is used along with the chemical inhibitors, it can rescue this acetylated. It can rescue the acetylated SMAD2 and SMAD3. So so from this immunostaining, they confirm the results, and in order to further validate the results, they also did the Western blot, and they can see that when the uh, chemical inhibitors, that is ETO plus acetate, is used during the in the definitive endotherms, they can see that high amount of acetylated or acetylated SMAD2 and SMAD3 proteins compared to the case where chemical inhibitors are used, or compared to these embryonic stem cells. And thereafter they did the mutations of this point base mutations of this SMAD2 and SMAD3 and uh, decided to check whether it's the SMAD2 and SMAD3 localization to the nucleus that affects their results or not, or that affects the differentiation or not. So when they did the when they prepared the mutations of this SMAD2 and SMAD3, for the SMAD2 mutation they did not find any changes in the nuclear localization. So however, for the SMAD3, the mutants has a reduced nuclear localization compared to the wild type SMAD3. So they decided to proceed with this SMAD3 mutant, that is K19 is mutated to R. So in this case they check the overexpression of the SMAD3, which is tagged with this flag, and they can see that overexpression of the SMAD3 is SMAD3 is overexpressed compared to the control in this wild type and K19R, that is wild type, and the mutant SMAD3. The SMAD3 can be overexpressed. So after validating that the SMAD3 are overexpressing in the cells, they next had to check whether this SMAD3 mutants localization, SMAD3 mutant localizations, SMAD3 mutants will prevent the will prevent the acetylation of these SMAD3 proteins, and thereby they will not be able to localize into the nucleus. So they decided to check this hypothesis whether the SMAD3 goes into the translocate into the nucleus and then helps with the differentiation or not. So in this case, when they do the immunostaining of the wild type SMAD3 and this mutant SMAD3, they can see that in the wild type SMAD3, the amount of in the wild type SMAD3, the amount of this flag, that is acetylated amount of flag or acetylated SMAD3 are reduced compared to the case where in the case of this wild type, the acetylated SMAD3 are high compared to the case of this SMAD3 K19 mutant. That can be seen here. In this immunostaining figures, it is not clear. However, when we the results of this immunostainings into the quantitative forms are present in the supplemental document, there we can clearly see that there is a decrease in the SMAD3 acetylation, SMAD3 acetylation, that is translocate into the nucleus in the mutant case compared to the wild type case. And when they check their difference, whether it affect the differentiation or not, the the problem in this SMAD3 mutant nucleation affects the differentiation of this human embryonic stem cell to the definitive endoderms or not. They then check the markers for the definitive endoderms, and they can see that there is a reduction in these markers such as FOXA2, SOX17, CXCR4, and along with that, they also see the reduction in those markers in the flow cytometry also. That is in the case when the mutants are used. SMAD3 K19 mutants are used, the amount of markers, definitive endoderm markers, are low compared to the case of the SMAD3 wild type. So, and these things are quantified in the panel that is on the right side of the panel G, where we can see that there is a significant difference between the gene markers of the definitive endoderms expressed in the mutant and the wild type. However, there is not significant difference between the gene markers of the definitive endoderms in the control in the wild type. So all this data indicates that is the SMAD3 nuclear localization, acetylated SMAD3, that helps in the translocation of this SMAD3 proteins into the nucleus, that results in the differentiation of the embryonic stem cells. So this is the final outcome that they have found out from this paper. So I hope you understand that the fatty acid oxidation pathways that is upregulated, that helps in the DE differentiation, and how these fatty acid oxidation pathways affect, that is, it will increase the acetyl-CoA within the cells, and this acetyl-CoA will cause the acetylation of this SMAD3 proteins, that leads to the translocation of them into the nucleus, and that helps in the differentiation of the embryonic stem cells by inducing epigenetic changes. So I hope you understood this paper and all their experimental results in brief. If you need to understand in detail how their experiments are conducted and their supplementary results, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.